So exam 5, it is chapters 9 and 10, everything through what we finished last time, and then whatever we cover today. So it probably won't be this entire chapter. My guess is it's going to be more of like the structure part about nucleic acids. But still, if you have less than an 80% in the mastering assignments, make sure that you do all of the questions for chapter 11, all that you can do, because all that's going to do is help to boost that mastering grade. So just keep that in mind as we go along through. So first off, to kind of make like a connection between the nucleic acids and all the other nutrients that we have talked about. Okay, so before we actually go into their structure, we have talked now, this is sort of like the last of the biomolecules is what they call them. So these are the large molecules found in the body. So we have everything from the carbohydrates to lipids to proteins, and now we have the nucleic acids. So those are kind of like the 4 categories. But if we were looking at size-wise, largest to smallest, or smallest to largest in this list, the smallest of the biomolecules are your simple sugars, right? So this is stuff like glucose, things like glucose, fructose. Glucose, remember, being your blood sugar. Glucose is only 6 carbons, 12 hydrogens, 6 oxygens, so it's a relatively small molecule. Increasing in size, the next larger is going to be your lipids, those fats and oils, the things that help make up membranes. So those lipids are going to be bigger than the sugars, but smaller than the other biomolecules we've been talking about. So if you remember, like fats and oils, and then phospholipids. That lipid barrier of the cell membrane. Getting even bigger, though, that's when we start talking about proteins. Now, not all proteins are bigger than lipids, but most of them are. Most proteins are more than 100 amino acids. So that means it's more than 200 carbons minimum in size. So they're much bigger. There are some, like insulin is a small protein, but most of them are larger. So think of those enzymes. As an example. But now we get to the first of the nucleic acids. So there is a nucleic acid that's called ribonucleic acid, or RNA. RNA is actually made of, instead of talking about like 200, 300, 400 carbons, minimum RNA is going to be in the thousands of carbons in size. So it's larger than proteins. I think about it as being just a little bit smaller than most of the starches. So remember, the polysaccharides are complex carbohydrates. Polysaccharides like starch, cellulose, fiber. Remember, those can be thousands of glucoses, and every glucose has 6 carbons. So here we're talking about like 6,000 carbons to 10,000 carbons. So they can be very large molecules. But far and above, almost like head and shoulders above all of the rest of them, is DNA. So deoxyribonucleic acid, that stands for DNA. So the D for deoxy is the D in DNA. This is like, I'm really going to like bust this pen before I'm done teaching this class. Okay, but that's where the name comes from, is deoxyribonucleic acid, is the DNA. It is the largest. So here we're talking about tens of millions, okay? Tens of millions, and even the small strands of DNA, the smaller chromosomes. So we are talking about millions, even billions of carbon per molecule. So these are huge. So nothing else even comes close to the size of DNA. So DNA and RNA, they're generically called the nucleic acids. And just as a general rule, a general sort of like definition, you always say your nucleic acids, this is your genetic material. So what they mean by that is your nucleic acids are established when you become a single fertilized egg. So when mom's egg, dad's sperm, when they join and unite, you end up with a set of DNA that is unique to you. You're part like your mom, part like your dad, like certain genes co-combine in just the special fancy way to make you. But you're unique and different from your parents genetically. And every single cell that comes from that single fertilized egg, which would be everything that it is what you develop into, all of that is going to be genetically different than anybody else, unless you're an identical twin. Okay? Because identical twins, that single fertilized egg grows and grows, and then it splits into two people. And then those grow. And so they are the most alike. There might be some changes in their genetic material, but it is a very, very, very small percentage. Okay? So they are the only ones that you could really say, yeah, it'd be very hard to tell you from your identical twin. But it's not hard to tell you from your siblings, even though you both have the same parents, which is amazing to me. But when that egg and sperm combine, you get a unique combination of this genetic material. And that is why you don't look exactly like your siblings. And I always tell people, like, my older child, she looks just like me. Okay? She does. Like her baby pictures and my baby pictures side by side, people are like, oh, that's Mary. I'm like, no, that's me. Or, oh, that's you. No, that's Mary. Because we really do. Like, we really have very similar features. My younger daughter, she looks just like her father's mother. So like her grandmother, she just, like, when she was born, I was like, she just looks like Grammy Mary. She looks just like her Grammy. I was like, and she still does. So even though same parents, every egg and sperm has a new potential or a different potential genetic combination that you can end up with. That's this nucleic acid. And this is going to determine what you look like. It's also going to determine, like, your characteristics. It can even determine some of your disease potential, like what you might evolve or develop over your lifespan. So DNA, first off, deoxyribonucleic acid. One, it is in the nucleus of the cell. So if you think of the cell, and if you remember back to when we looked at, like, the cell with the phospholipid, so you remember that the cell looks almost like it had this thing in the very center. So if this is the cell, this is the nucleus. So it is kind of a central organelle, and this is where the DNA is found, and this is where it stays. So what is special about DNA is that it contains what are known as genes. The sequences found in the DNA, certain segments, sections, areas of the DNA, the sequence of the DNA contains the information that your cell can use to make a specific protein. So hemoglobin, there is a section of DNA, and the sequence of it tells the cell how to make hemoglobin. Same thing with collagen, the other big abundant protein. So collagen is a sequence of DNA that can be used by the cell in order to build collagen. It's estimated that there's about 20,000 different genes contained within the DNA. So we're talking about 20,000 different proteins. Some proteins, remember we talked about proteins in the last chapter, some proteins might help to build antibodies. Some proteins might be carriers like hemoglobin. Some might be hormones. Some of those proteins might be the receptors or those channels. So all of those different functions of the proteins we talked about in the last chapter, that's where these 20,000 genes come from. Different proteins that different cells need. Different cells are only going to use certain genes. So best example, hemoglobin. Hemoglobin is made by what cells? What cells make hemoglobin? What cells are filled with hemoglobin? Yeah. And that's the only cells that make hemoglobin. Even though the hemoglobin gene is in all cells, the only cells that use it are red blood cells. So that's super specific. Same thing with cells, like cells that have to maintain tendons and ligaments. They're the ones that make collagen. Red blood cells don't need to make collagen. They're just concerned with making hemoglobin. So different cells use different genes in order to make different proteins. And it really depends on what job the cell has to do. So let's talk about the building blocks. So the building blocks, just like amino acids are the building blocks of proteins, nucleotides are what they call the building blocks of nucleic acids. A nucleotide has three things. A nucleotide has a sugar, and it's a 5-carbon sugar, so remember that makes it a pentose. That is a sugar with 5 carbons is a pentose. It also has a phosphate group, so remember that the phosphate group is PO4. So whether it's DNA or RNA, it has a sugar and it has a phosphate. It also has what they call a nitrogenous base. So sometimes this throws people. Well, a nitrogenous base is called that because it has the ability to accept protons. So remember we said that acids are proton donors, bases are proton acceptors. So a nitrogenous base, the only reason they call it that is because if there's extra hydrogens floating around, it can take those on. So that's why they call it a base. These nitrogenous bases are different, though, whether you're talking about DNA or RNA. So here's the differences, and I don't expect you to remember the structures of them or being able to identify which one's which, but you should know the 5 names, and you should know which is RNA-based and which is DNA. So RNA and DNA have these 3. RNA and DNA have adenine, guanine, and cytosine. Okay, so A, G, and C. Adenine, guanine, and cytosine, these are found in DNA and they're found in RNA. The only difference with the nitrogenous base types is that DNA has thymine, which is the next one, THY, not thiamine, that's actually a B vitamin, but thymine, THYMINE. And thymine is one that you find in DNA. RNA has uracil. And if you look at the two, can you see where they're different? There's only one thing different. What's that? What's a CH3 group called? Does anybody remember? In organic? If you have a CH3 group hanging on, that is it's a single carbon hanging off. A CH3. Remember, like a CH2, CH3 is an ethyl. This is a methyl. That's the only thing. Notice that the thymine has that CH3 group. Uracil doesn't. But other than that, do you see that it's exactly the same? So that's really, it's not like, ooh, it's huge and different. It's kind of like, they're very similar, but this one is DNA only. This one you only find in RNA. So this is one of the differences between DNA and RNA. Both of them have A, G, and C. Adenine, guanine, and cytosine. But DNA has thymine and RNA has uracil. The other difference number 2, DNA has the sugar called deoxyribose. RNA has the sugar ribose. And again, only one difference. Look at carbon number 2. So in DNA, deoxyribose is missing an oxygen on carbon number 2. In ribose, in ribonucleic acid, its sugar, ribose, has that oxygen on carbon number 2, which makes it an alcohol. So notice here and here. That's the only difference, really. So that's where the name deoxy, because D means to remove. So deoxy means it's one less oxygen. So that's kind of where that name comes from. So DNA doesn't have an oxygen on carbon 2. RNA does. But other than that, notice that these are both pentoses, both 5-carbon sugars. Very similar in terms of their shape. The way that nucleotides are connected, whether it's DNA or RNA, is the same. So this is a nucleotide. So here's one nucleotide, and here's another one. Right? So the nucleotide, see that it has a phosphate, a sugar, and a base. they form a connection, they form a link, just like amino acids get linked together into a protein, just like sugars get linked together in a polysaccharide. It is a condensation reaction. So there's an oxygen that is pulled off of the phosphate and a hydrogen that is pulled off of the sugar. There's also another hydrogen floating around because it's right here. There's a hydrogen floating around in the solution that ends up having to be the extra hydrogen. But you see that water is produced. So it's still considered a condensation reaction. Always think condensation reaction forms water. And it acts to join molecules together. So this joins the nucleotides. So the sugar links to the phosphate by a condensation reaction, then the next sugar down links to a phosphate with another condensation. So you begin to have a chain formed where you have the connections between the sugar of one nucleotide and the phosphate group of the other. So it starts to look like this. So in this, do you see that this is 4 nucleotides linked together? So this, kind of like we did with the amino acids in the last chapter. So do you see each one has a phosphate with the sugar? Hanging off of the sugar is the nitrogenous base. Then there's a phosphate sugar linked to a phosphate sugar linked to a phosphate sugar. So the top one, where you write out the word phosphate, write out the word sugar, you can simplify it by just using a P. So the P in this part right here, this is just showing you that it is, like we can take phosphate and just shorten it to an S because it's a little faster to write. We can take the S, sugar, and just shorten it to an S. So we got a phosphate to a sugar to a phosphate to a sugar. On one end is the beginning. Do you remember in proteins, I said nitrogen's on one end and there's a carbon with 2 oxygens. So there's kind of a beginning and an end. Same thing happens with the nucleic acid. One end is going to have a phosphate. The opposite end will end with a sugar. So there still is kind of a direction. Think of it like a sentence. So it's not exactly the same in either direction. So typically you start with the phosphate end and you go towards the sugar end. So P, S, P, S. Notice that that kind of looks like the backbone of a protein, right? Remember how we wrote it NCC, 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 and looking back in chapter 10, same thing. P, S, P, S is just phosphate sugars linked together. Notice that the bases always hang off of the sugar. They never hang off of the phosphate. They're always attached to the sugar part of the nucleotide. So the bases is the A, G, C, and T. Adenine, thymine, cytosine, and guanine. And this is the same with RNA. So when you link nucleotides of RNA together, phosphate links to sugar, links to phosphate, links to sugar to make that long chain. And then you have the bases hanging off of the sugars, except that G, C, A, and U are the bases that you would find. So RNA would have a U, not a T. So how did they figure some of this out? So there was a lot of research that went on, and this was like in the very late 1800s, early 1900s. One guy that worked on this, his name was Chargaff. What he did is he took cells, he isolated DNA, and he looked at the amount of adenine and thymine, cytosine, and guanine that you found within cells. So he looked at like frog DNA, mouse DNA, human DNA, plant cell DNA. So he just kept on collecting different samples of DNA and evaluating what are the amounts of A, T, G, and C. So one interesting thing was all living things have A, G, T, and C in their DNA. The other interesting thing that he found, though, was that relatively the amount of T was always equal to the amount of A. So there was a ratio of them. So if I had X amount of T, I'd have X amount of A. If I had 200 molecules of G, I had 200 molecules of C. So they were equal. So he found that C and G were equal to each other and T and A. So he said, well, that can't be random because too many different living cells contain this. So there has to be a relationship between A and T. There has to be a relationship between C and G. So a British chemist, Rosalind Franklin, she was earlier in the 1900s. She actually took strands of DNA and froze them and did what they call X-ray crystallography. So she took strands of DNA and turned them into crystals. So made them solid and then ran X-rays through them and found that DNA had a helical shape. So when she did her X-ray crystallography, she saw that it created this like spiral. So she was like, So she published that. Chargaff published that. They didn't really talk to each other so much. And so then here comes Watson and Crick. So Watson and Crick, James Watson and Francis Crick, this is in like 1950 now. So these people did these experiments in the early 1900s. People were talking about it, but they didn't really know how it was put together. So Watson and Crick, and they didn't really do a lot of the research, but they took everybody's information and they put it all together like a puzzle. So there they took Chargaff's information, they took Rosalind Franklin's information, Linus Pauling had done some research. So they pulled all this together and they sat around and they said, okay, we have a helix, we have A and T, they've got to be equal to each other. G and C have got to be equal to each other. So they said DNA must be a double-stranded helix. And whenever there's A, there's going to be a T on the opposite side. And wherever there's a G, it's going to match with a C. And that's how they even out or how they're equal according to the number of bases that are found. So they found that when they started looking at the chemistry of it, they found that if you had them lined up side by side this way, the A and the T, they didn't fit together. Their chemical structure would actually repel each other. But if they took the two strands and they flipped them, so that one strand ran sugar phosphate, the other strand ran opposite, sugar phosphate in the opposite direction, they found that then they, instead of being this way, they fit together perfectly and they can hydrogen bond. So they found that the bases, the two bases that pair off A actually forms two hydrogen bonds with T, and G can form 3 hydrogen bonds with C. And remember we said hydrogen bonds are pretty weak, but when you have a lot of them, they become much more significant. This is pretty amazing that they did all that they did with the technology that we had at the time, because we are talking about DNA. DNA has 3 billion nucleotides, 3 billion pairs of bases that exist in that double strand. And they were able to come up with this structure and this arrangement with something this big. So they won Nobel Prizes for this, even though they didn't really do the research. And the people that did the research didn't get any Nobel Prizes because they published all of this first. And then all of the research afterwards all said, yes, this is exactly what it is. So they were kind of an interesting pair. They would literally go to the pub and sit and drink beer and talk about this stuff. Okay? So they were really good at just putting stuff together, not so much in the experimental part. Okay? So they found now we know that adenine and thymine are going to hydrogen bond, which is why they're equal in amounts. Guanine and cytosine can hydrogen bond, and that DNA contains a double strand. The two strands look like this. So the purple little strings, looks almost like ribbons down either side, those are the sugars in the phosphate. So Wherever there's a nucleotide, there's a sugar. And then in between the sugar is where you've got a phosphate. So this is a sugar, then there'll be a phosphate, then a sugar, and then a phosphate. Okay? So everywhere there's an organic base, A, C, G, or T, you've got a sugar attached to it. In between those sugars is a phosphate that's tucked in. Looking at A and T at the top, do you see the hydrogen bonding? Do you see how there's a nitrogen on adenine that has a hydrogen, and it can hydrogen bond with the oxygen that's on the one across, on the thymine. The oxygen across, so it's like the nitrogen and oxygen share that hydrogen up here. That's a hydrogen bond. Same thing in the one below. So the reason that it can form two, adenine has a nitrogen, thymine has a nitrogen with a hydrogen, so they like share the hydrogen between them. That creates the hydrogen bond. So you'll notice everywhere there's an A and a T, you see two sets of dots connecting them in the strand. Everywhere there's a C and a G, cytosine and guanine, there's 3 hydrogen bonds, and you can see the 3 hydrogen bonds in this bottom structure. So this is guanine and this is cytosine, and when they're set together, they hydrogen bond because they're very complementary in structure. So DNA, double-stranded, forms a twist, a helix because of this hydrogen bonding, stabilizes it, has A, C, G, and T. Those are those organic, the nitrogenous bases that they have. A, hydrogen bonds with T. C, hydrogen bonds with G. And that's what helps hold them together. And we're talking about 3 billion nucleotides. So if you took the DNA in one cell and you could lay it end to end to end, the DNA in just one cell would actually stretch out about 6 feet in length. So like just think about like one single cheek cell, and you took the DNA out and you could like tease it all apart and stretch it out. You'd be talking about something that's this long. So when you have strands that are this long, the odds are they can get twisted, they can get wrapped around each other. They might end up getting like knotted together. And if they got knotted together, then that would be very bad, because then if the cell had to divide, they'd be all torn. So then this genetic information would get destroyed. So the way that the DNA actually arranges itself is it forms what are called chromosomes. So when the cell divides, it takes its DNA, which is the double-stranded helix, and it wraps it around these really small proteins called histones. So histones are positively charged, and they are attracted to the phosphates of the DNA. So remember that phosphate, PO4, is a 3-. So the phosphates in the DNA are negatively charged. So you know opposites attract. So those histones help to hold the DNA. And I think of this as being kind of, it spools the DNA much like you would spool thread. So like if you go buy thread at the store, you don't just buy like a big long string. It's all nice and spooled. You get 300 yards, you come home, you're good for a while. Okay? If you had 300 yards of thread and they just gave you a big pile, you would never get it untwisted. It would end up being knotted. It would end up having little connections. You'd be like, forget it. That's really what this does. Histones create a spool of DNA. So the DNA goes from this long to being microscopically long. So it shortens about 1,000,000-fold. And instead of being super, super thin, it helps to thicken it about 1,000,000-fold as well. So we're going to shorten it and thicken it so that now it's like we have little parcels. We have ways to separate the DNA much easier, and it doesn't get knotted, it doesn't get torn, it doesn't get twisted, it doesn't get damaged. So they call this like the tertiary structure. So this, remember in proteins we had primary, secondary, tertiary structure? Well, the primary structure of DNA is just the nucleotides. the order of the nucleotides. The secondary structure is the fact that DNA forms a double-stranded helix. Tertiary structure is this hyperfolding that DNA takes in order to help keep it stable so that it doesn't get damaged. So how's RNA different, right? So if we know how DNA is, so then tell me, how is RNA different? So DNA is double-stranded. This is a really good kind of compare and contrast. DNA is double-stranded, has a deoxyribose sugar. The bases are ATGC. It's 3 billion nucleotides, so you got to put all those commas in. 3 billion nucleotides in length, and that's in just the nucleus of a single cell. RNA, though, is just a single strand. So RNA doesn't form that double-stranded helix. It is just a sugar-phosphate chain hanging off, or the sugar is ribose instead of deoxyribose. And the bases have that one difference. Instead of T, there's a U. RNA is much smaller. RNA is typically only 500 to 5,000 compared to 3 billion, so much smaller in size. RNA is actually like the DNA of just one topic. So if you think of the DNA as being like Wikipedia, the DNA is like an encyclopedia. It has all of the genes to be able to make all the proteins that all the cells in your body will ever need. Well, RNA is just the information for one protein. So RNA is made from a gene in the DNA. So you can almost think of RNA as being like a short form. That is why it's so much shorter in terms of its length. It's only made from a specific sequence of DNA, and then it can be used to build a protein. So this part that they have down here, this is kind of like describes how proteins are made in the cell. So in the nucleus, you have a sequence of DNA called a gene. And the sequence of DNA, that one specific gene, we can make a short version of that, which is called RNA. The step that does this is called transcription. So this process is called transcription. So all we're going to do is take one sequence of DNA and convert it to a sequence of RNA. This happens in the nucleus. So transcription happens in the nucleus because that's where the DNA is. Once the RNA is made, now the RNA can get moved out of the nucleus of the cell and out into the cytoplasm of the cell. So the cytoplasm is the larger sort of like interior of the cell. And in the cytoplasm is where you have all of the machinery to be able to take that RNA and use it to form a protein. That step is called translation. So transcription is the first step where DNA is converted to RNA. It happens in the nucleus. Translation is the second step where RNA is converted to proteins, and that happens in the cytoplasm. So we'll go through, we've talked about all of the parts of DNA, right? So DNA, 3 billion nucleotides, the certain sequences in the DNA make up genes. But now when we make RNA, there's actually more than one kind of RNA that you can make. So when you make RNA, you can make what is called messenger RNA. And they oftentimes just use a lowercase m, so they just put mRNA. That stands for messenger RNA. This is what's actually made from a gene. So messenger RNA is what's made from a gene. In order to take that RNA and make it into a protein, you have to make a ribosome. So ribosomes are extremely small organelles that are found in the cytoplasm. Ribosomes are made up of protein and RNA. So the kind of RNA that they have, they named that and said, well, it's different than mRNA, so we're going to call it rRNA. So this RNA is made to help form ribosomes. So that's why the R part of it. And it's these ribosomes that actually build proteins. So they're really the protein factories, the protein synthesis factories. They're the ones that take two amino acids and put them together. So they're kind of like an enzyme inside of the cytoplasm of the cell, and their job is to link amino acids in the right order. So that's the job of a ribosome. Then the third kind of RNA. This third kind of RNA is the RNA that is responsible for bringing amino acids to the ribosome. So if the ribosome's got to link amino acids together, where do they come from? They don't just like randomly float by. There's actually an RNA form that brings them, that specifically brings them into the ribosome, and they called them transfer RNAs or tRNAs. So a tRNA, its job is to carry amino acids to the ribosome so the ribosome can put them together. So it's all based initially on the sequence of the DNA. So if we look at the steps of transcription, so remember I said that this is the first step. So if we go from DNA to RNA to proteins, this is the first step. So the gene, which is the sequence of DNA, has to be used to make a strand of messenger RNA. So the easiest way to show you this is to show you it as a picture. So in this, the two purple strands, this is DNA, right? So remember it's the double-stranded helix, so they kind of have it drawn like a helix. So it's sort of twisted. So it's got two strands. One strand contains the sequence that's the gene. The other strand is just there because it's a double-stranded helix. But the one for this one, the bottom strand is going to be our strand that is carrying the information of the gene. This big gray thing that looks like a big bubble kind of covering everything, that's the enzyme that does it. So there's an enzyme in the nucleus of the cell that actually binds to DNA and uses one of the strands of DNA in the gene to be able to build that blue strand. The blue strand is your mRNA. And you notice wherever there is a G on the DNA, what will the RNA match? A C, right? Because G and C, they hydrogen bond, they're complementary to each other. So if on the DNA, like in this example, there's a G, then my mRNA is going to be a C. And these can hydrogen bond. So that's how you know you're putting them together correctly, because they complement each other. So then the next one, notice, is also a G. So then the enzyme's going to put what next to it? A C. So it'll link it and add another one. The next one is a T on the DNA. So if we go along, do you see the next one's a T? So what will it match? An A. So the enzyme adds an A, links it together, right? It's just doing a condensation reaction. It's just linking RNA nucleotides together. Next one again is a G, so it'll put in a C. Link it together. Sorry, that's a C. That's a G. Then a C, links together, links the G. So if we're just following along, but now these two, the next two are A's. So the next two, instead of being T's, it's going to match the U. So do you notice that on the DNA, if there's an A, it matches a U. Linking them together. Next one's a T, it would be an A. So the U is like kind of marks that RNA as being RNA, not DNA. So this is what the enzyme does. It just goes along down. It uses one of the strands of DNA to be able to synthesize a complementary strand of RNA. And in fact, there is a part where there's a stop and a start. Like there's a beginning and an end. So it's sort of like in a sentence. So there are parts of the gene. At the beginning of the gene, there's a sequence that tells the enzyme, start here. So that tells the enzyme, pull the DNA apart and start making an RNA strand. Then there's a sequence at the end of the gene that says stop here, kind of like a capital letter and a period on a sentence. So there is like kind of like instructions by the sequence of the DNA. So it keeps on going until it gets to the very end. When it gets done, so at the end of the gene, remember that the gene is the DNA. At the end of that sequence of the gene, the enzyme lets go of the RNA. So it's made in the nucleus, but as soon as it's made, it doesn't stay there. It actually moves out of the nucleus and into the cytoplasm. So we can do this one. Okay, so here's a DNA gene. So remember, DNA is double-stranded. So when you look at it, you see, oh, there's a top and a bottom. But we're going to say the bottom strand is the one that's used. So you tell me, we'll just go straight across. If you have a T, the mRNA will be what? A. A will be U, right? And then C is G, then T, A. A is U, then C, G, U. U. Then 4 U's, because those are the matches to the A's, right? So I'm just looking at that bottom strand. Anywhere there's a T, you put an A. Everywhere there's a C, it matches a G. G matches C. But everywhere there's an A, you match a U. Okay? So this is my new strand of mRNA. So T is A. A, C, C, C. 4 C's in a row, then 2 U's, then a G, then an A. Right? I didn't make any mistakes, did I? Let me do the first time. Yep, so then this one is the 5. So now, because they're backwards, right? Because they go the opposite direction. So the sugar end, phosphate end, becomes the phosphate end and the sugar end. But this is just an RNA strand. Now this goes to the nucleus, goes out to the cytoplasm. Because in the cytoplasm, that is where the ribosomes are. So the ribosomes are what need this. They need this sequence of RNA in order to make the protein. So this is really transcription. Okay? The enzyme finds the gene, unwinds the DNA, and it makes a strand of RNA from one of the DNA strands. And that's why I say it's kind of a short form, because it's not the entire sequence of DNA. It's one specific little area of the DNA sequence that codes for a certain gene. So now we go out into the cytoplasm, and here is where your rRNA and tRNAs come into play. So here you can see all 3 RNAs. So here is what we just made. There's the mRNA, the messenger RNA. That's what we just made from the gene. So that's that blue strand, right? That's coming along here. The thing that looks almost like a hamburger bun, top of it's purple, bottom of it's a little darker blue, that's the ribosome. So the ribosome is these two pieces, and that's why I say the meat, they look like a hamburger bun, okay? Because there's a large subunit and a small subunit, and they like sandwich the RNA. And then they're going to start to move down the RNA by using those nucleotide sequences. Because they want to use the nucleotide sequence on the mRNA to be able to match the right amino acids. So the things that match the mRNA are the transfer RNAs. So I always think of tRNA, it looks almost like a strange bent up hanger, like a wire hanger. So it's kind of got like this twistiness to it. But there's 3 bases on the tRNA that will complement the sequence of the mRNA. So notice here, notice where I have a G on the mRNA, the tRNA will have a C because G and C can hydrogen bond. Wherever I have a G, there's a C, and where that I have an A, I'll have a U. So we'll have hydrogen bonding here, here, and here. So that allows those to fit together. So you can think of them like they come in and they sit down. They have to, all 3 of those have to match. If one doesn't match, then it's like this. If one doesn't match, then it's not going to be uniform and it won't be able to fit into that spot. But the right tRNA fits, that's the amino acid that belongs there. The tRNA is carrying an amino acid. So the tRNA brings amino acids to the ribosome matches the mRNA sequence. So once you have one, when the ribosome begins to move, the ribosome always slides down the mRNA by sets of 3. So you see how this is in a group of 3, that UGC and the ACG, that set of 3? It's always done as 3. They call this 3-base sequence a codon. So this order, ACG on the mRNA, that determines what the amino acid is that matches. So how do you know which amino acid it is? Well, there is what they call the genetic code. So they have actually figured out what sequences on the mRNA will determine what amino acids belong in that position. So now you've got to remember back to some of those short versions, or at least recognize this. So do you see like PHE and LEU and ILE and VAL? So do you see these? These are the 3-letter abbreviations for amino acids. So like, I don't know if you remember, we talked about like valine, that's VAL. Leucine is LEU. Lysine is LYS. So you don't have to remember those, but just those 3 letters, that stands for a certain amino acid. So if on the mRNA, my sequence is UGU, that will match a CYS carrying amino acid, which is cysteine. So this is the amino acid that fits in that position. So if there's a UGU on the mRNA, then it'll be the only tRNA that fits that spot will be one that's carrying CYS or cysteine. So let's look at those ones here. So this is that sequence I just copied down a few minutes ago, okay? This is the exact same one, the one that I went through and we wrote. So let's put it together. So here's our mRNA. And we're going to start at that 5' side because that tells the ribosome start on this end. So if the ribosome comes down and sits here, so the ribosome is this like little hamburger looking thing. So the ribosome sits here, and see where there's AUG? The AUG is going to only allow a tRNA that matches that AUG. So it'll be a UAC. But if we look back on that genetic code, find AUG. So do you see it in the first column? So AUG, that always codes for MET, which is methionine. But notice always all proteins start with AUG. So that is also like another signal to the ribosome. This is the beginning. So all sequences, all mRNA strands at the beginning of the gene, it'll end up having an AUG. So if we go back, that means this amino acid is going to be an methionine. So this is the amino acid that corresponds to the AUG sequence. So once this is attached, this is a signal for the ribosome to move down 3 spaces. So it's going to go to here. Now you got to go find AUC. Do you see that AUC is the next one? So here's our little bent up looking tRNA. We have AUC. What's the amino acid that AUC carries? Do you see that's like 2 up from the other one, still in that same group? So it's going to be an ILE. So now I have methionine, MET, sitting right next to another amino acid. So there's like the nitrogens and the carbons of the amino acids are side by side. A condensation reaction occurs so that these two get linked together. That's a peptide bond. And the ribosome does it. So there's now a peptide bond that is formed between these 2 amino acids. Now I have 2 amino acids linked together. As soon as that bond is formed, this tRNA link is broken. So it's sort of like transferred. So amino acid 1 gets linked to amino acid 2, and the tRNA of amino acid 1 gets dumped out. And as soon as that happens, the ribosome moves down 3. So now we're going to start this like repetitive process. We're going to go down 3, put our tRNA, find that sequence GUG. Okay, so if you go forward 1, GUG is here. So do you see GUG? It's going to carry the amino acid VAL, which stands for valine. VAL is the one that it's carrying, and that's going to put these 2 side by side. Once you have 2 amino acids side by side, the ribosome is going to link them together. That's a peptide bond. And then it'll break the link to the transfer RNA that was carrying the first one. Then ribosome slides down 3. It goes from, now it's got GGG. So what will this amino acid be? GG, yep, that's glycine. Oh, this one, not GGC. So do you see that one down in the corner? GGG. It's going to be carrying, the tRNA is going to be carrying GLY. Then what are we going to do? Link them together, right? Remember, you're going to link them. So the ribosome's going to match a tRNA, link the amino acids together, breaks the connection to the back tRNA, ribosome slides down. So this is just going to continue. So in the next one, I'm going to slide down. So the ribosome moves down here. Now I got CGU, which is arginine, ARG. Ribosome, if you go forward, find CGU right here. Do you see it on the chart? So CGU is carrying the ARG. So it's this one up here. It's carrying ARG. Once it has it side by side, ARG and GLY, peptide bond is formed. So we're linking this one, breaking this one. Sliding down 3. Just try to keep changing colors each time so you can see it maybe. Going down 3, UGU. UGU. Yeah, CYS, which is cysteine. I don't expect you to remember the names of them. And I wouldn't expect you to actually CYS, links them together, breaks this connection. So now really right now, I have a cysteine connected to an arginine, connected to a glycine, connected to a valine, connected to isoleucine, connected to methionine. Like, so I have like all these amino acids that are now like starting to like come out of the ribosome. So it looks almost like a little string, and it gets longer each time you go through this one step. I do not expect you to memorize the names of amino acids. I would not expect you to just randomly know which one matches. If I gave you this, if I gave you an mRNA sequence and I gave you this, could you go every 3 and figure out what the amino acids are? Remember, it's always going to be 3 bases on the mRNA strand. So it's always going to move down each turn. UGU, we go to UUU, 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 PHE, phenylalanine. See, that one's in the upper left corner. So PHE is the amino acid it is carrying, PHE. These get linked, this gets broken. Ribosome slides down 3, matches the tRNA, carrying a certain amino acid that you can figure out from the genetic code. Forms a peptide bond. ACC, here, yeah, which is threonine. ACC is T, it's carrying THR, stands for threonine. Links these together, that one falls off. So now all the ones with X's are gone. They like fall off the back. The ribosome just kind of keeps on chugging along. So these little empty tRNAs fall off. But attached to threonine is 1, 2, 3, 4, 5, 6, 7. So there's 8. So there's 8 amino acids now in our chain. 2 more rounds. So we go down 3. And we have CCU. CCU stands for what? Proline. PRO. These get linked. This gets broken. Now we're at the end. UGA. When the ribosome slides down, it comes into a problem. Because UGA, when you look at this, what does this stand for? So it doesn't actually code. There is no tRNA that fits here. This is called a stop codon. So the codon is the group of 3. So UGA, a sequence on the RNA of UGA, tells the ribosome this is the end. Stop here. Just like AUG is the start codon, UGA and UAA, UAG. See that there's 3 of them? There are 3 stop codons. So if the ribosome gets to any of these 3, it will actually just release. So it's sort of like just stops and lets go. So the big the hamburger comes apart, lets go of the mRNA and releases the new protein. So that's how proteins are made. Now, you think like, God, this sounds like it'd take forever. It really does. To me, it's like slide down, match, form a bond, slide down, match, form a bond, slide down. But in fact, this is an enzyme. So remember that enzymes help to speed up chemical reactions by lowering that activation energy. So this, they say that this, like a ribosome, can actually link amino acids together, hundreds of amino acids in a second. So it can just like literally goes right down the mRNA and like links them. As long as you have amino acids carried by tRNAs, there's nothing to stop the ribosome. And if the ribosome gets to the end, It's an enzyme. It'll let go back to the beginning and connect and make more. So you can make lots and lots and lots of protein from one ribosome and from one mRNA, as long as you have enough amino acids. So remember, we were talking about getting enough protein in your diet. You have to have the building blocks. If you don't have the amino acids, you won't have enough tRNAs to carry them. You won't have enough amino acids on those tRNAs, and that'll limit how quickly cells can make protein. So you've got to have those building blocks in order to do it. This also requires ATP because we're building stuff, right? Remember, synthesis reactions are almost always energy demanding. So we're doing condensation reactions. So this is an ATP demanding kind of process. So then one thing I wanted to talk about, well, what happens if the DNA is wrong? What if the sequence is wrong? So that is what they call a mutation. So definition, a mutation is any change in the DNA sequence. That's what a mutation is. Mutations can be things like, so if there was an A and a T, now there's a G and a C in the sequence. We're talking about 3 billion nucleotides. That's a lot of pairs. That's a lot of sequences. So one question is like, well, how do mutations happen? Right? So how? How do mutations happen? What can cause a mutation? So one is they can be spontaneous. Mutations can just happen. And one of the times this happens is when DNA gets ready to divide. When your growing cells need to divide, they have to double their DNA. So that means they got to take 3 billion nucleotides and make 6 billion. And if you had to read 3 billion letters, do you think you would ever make a mistake? Yeah, I make a mistake when I'm trying to type 10 letters, okay? Backspace, backspace, backspace. Start again, backspace, backspace. I do it all the time. So you would think the cell, it does make mistakes. When the cell is trying to copy DNA before it divides, it might make a mistake. So they call that spontaneous. So these are like random errors that can occur. But other reasons that you can have a mutation is because of things called mutagens. So mutagens are materials, environmental agents that can cause a mutation. One of those things are chemicals. Chemical mutagens are things that are the sides. So if you think of a pesticide, an insecticide, an herbicide, anything that is a side, what is its goal? Like, what does a pesticide do? Destroys or kills a pest. Herbicide destroys plants, like Roundup. So Roundup is an herbicide. So if you don't want any weeds growing between the little cracks in your driveway, you spray Roundup, right? And they turn all brown, they go away because it kills plants. It's an herbicide. So what is an insecticide? Kills insects, right? So if those blasted fire ants in the yard, so you go put those little insecticide granules on there and just hope that they come and carry it down into their little pile and then they all die and leave. Because the worst thing is when you stand in them and you don't notice it, and then suddenly they're on your shoes and they're on your ankles and they're just biting you and it's awful. But all of those pesticides, herbicides, insecticides, they are capable of destroying or killing living cells. So a lot of them, if you look, it says like harmful if swallowed. do not let it come in contact with skin. Wear gloves while handling, because if it can kill other living cells, it could potentially cause damage to ours. So any of the sides, you really want to be careful when you handle. You really want to wear gloves. You really don't want to end up like ingesting. So wash your hands after using things like this, just because there is a risk. Second, energy. So how many people have ever had a sunburn? Okay. Have you ever had a sunburn? You've never been sunburned? Wow, that's like, just because you have dark skin doesn't mean you can't get a sunburn, okay? It does mean you have a lot of melanin, which does give you protection from ultraviolet light, but you can still get sunburned. So ultraviolet light is energy from the sun, and ultraviolet light can actually cause sunburn. So if you don't go and wear sunblock, that is that high energy that can cause damage. In fact, there is an association with having repeated sunburns and development of what? When you get older? Skin cancer. Okay? Because this ultraviolet light can cause damage to skin cells and make some of those skin cells undergo mutation. And mutation can then cause cells to grow irregularly. So those are considered mutagens, chemicals as well as energy. The third one, though, is viruses. So viruses, which infect a cell, can sometimes insert themselves into the DNA of that cell. So if the virus goes into the cell and inserts itself into the DNA, that's going to change the sequence of the DNA. And so viruses have been found to be able to cause mutation. So there are viruses that are associated with creating mutation and potentially causing problem. So a mutation that is in a generic body cell, so mutations in somatic cells might become abnormal, but it's only going to affect that cell. So this would be like if you get skin cancer. Normally that's because of a mole, a melanocyte, a pigment-producing cell on the surface of your skin that ends up having a mutation and starts growing without control. And so then that mole gets really big, it gets really dark, it gets really irregular, it might be really raised and kind of unusual, and you can end up developing what they call melanoma. So that is a cancer cell that originated in that pigment-producing cell. Started off as a mole, but now it's gone out of control. But it's only right there. Yes, it might spread, but it came from that one cell. If it is a mutation that happens in a germ cell, germ cells are not bacteria. The word germ is referring to your sex cells. So sex cells are egg or sperm. So if you have a mutation in an egg and that egg gets fertilized, then what does that mean for that offspring? So like I said, if we're talking about a somatic cell, that is just like a skin cell, muscle cell, brain cell. So if you have a mutation, something will happen to that cell. But if you have a mutation in an egg cell and it gets fertilized, what does that mean about that offspring? Then that means that every single cell of the offspring will have that mutation because, right, we originated from one fertilized egg. So if there's a mutation in that original egg, then that mutation will be carried to all of the cells of that person, and then all of their offspring are affected by it as well. So this is the reason why we have different skin color, hair color, eye color, is because of mutations that happened in the germ cell layer. And so you had this family that all had brown eyes, and suddenly there was a mutation and there's a kid with blue eyes, and they're all like, but then that gets carried on to his kids. Okay, so that offspring, their children, end up having that mutation. That's how we ended up developing differences, is all because of mutation. So not all mutations, you always think of mutations are bad, right? I always think of mutations and I think of cancer, right? I think of a disease or something bad. Well, mutations really develop a whole lot of variation among a species. Sometimes they're beneficial, sometimes not so much. Having darker skin is beneficial because it protects you from ultraviolet light. Being super, super pale, that's not a beneficial thing because you're much more likely to get skin cancer because of multiple sunburns. So these are things that cause mutation, and then it's much more serious if you have a mutation in a germ cell. It's the thing to remember because that means that the offspring, all those cells of the offspring will have that mutation as well. So the next question, and I don't know why I don't have this in the right order. I apologize. So if there's a mutation, couple of things. One, the mutation might have no effect at all. So they call these mutations silent mutations. In fact, it is estimated that only about 3% of the DNA, those 3 billion nucleotides, only about 3% of the DNA actually codes for genes. The 97% of the DNA have other functions that actually help to make the DNA more stable, help to protect those gene regions from any kind of damage. So if there's mutations in the non-gene areas, that would be a silent mutation. It does just add to our genetic differences. Like I told you in the beginning, your sequence of nucleic acids is different than mine. That is because there have been all of these silent mutations over the generations and generations that haven't really affected how you function, but they have changed the sequence of DNA. So it was interesting. I took a class about 10 years ago. I took a class where there was 20 people, and all of them were from different racial backgrounds, okay? Some of them, there was a couple of Asians. There was a couple that had parents that are African descent, some that were European. So all these different, one was Australian. I will tell you that my mother is Native American. So my mother was actually born and raised on an Indian reservation. So both of her parents are 100% Native American. So when we did this DNA analysis to look at racial organization, even amongst our class of 20, I was the one that didn't match anybody. I was and when they were looking at it, they were because I kind of came out more like Russian, because they believe that Native Americans came across from Alaska and then came down into North America. And so I had some connection, but I didn't match anybody else in the class because of those changes. Because my Native American people lived in the Americas for thousands and thousands of years, not breeding or having children with any Europeans or any Africans or any other groups. So that means that group, that DNA sequence is different than all of these other groups. So it's really interesting just how that ends up applying. So there is differences if the mutation, there are some mutations that it wouldn't change the amino acid, some mutations where the new amino acid might still have the same polarity. So if you had a polar amino acid and it gets switched to another polar amino acid, that might not change the shape of your protein. But what if it did? So if it did, if we had a mutation that changed the shape of the protein, that might change the protein's function. So example, what if there's a change in the DNA, and that means that instead of having a polar amino acid, you get a non-polar amino acid? That, remember how the globular proteins, that could cause another shift in the twist of that protein, and that might affect how that protein functions, especially if that protein is an enzyme. Because remember how enzymes, their shape is so important to their function. Sometimes it can make proteins that are super short, maybe even make proteins that are longer than they need to be. So what can happen if this occurs? Well, one, the cell might die. If this is a really important enzyme or a really important gene, then that protein doesn't work and the cell might not survive. This could also, though, cause change in how that cell grows. So one of the jobs that DNA has is it helps to control what's called cell cycle, how often the cell goes through mitosis, when one cell divides into two cells. So the DNA does control that. So if you have a mutation in one of those genes, then you might lose control over how quickly a cell divides. So then a cell just begins to divide without control. That is what happens when you have a tumor. Some tumors spread, right? So some tumors stay in one little area. They just remove it. It hasn't gone anywhere. It's nice and encapsulated. But then there are some abnormal cells that can actually spread. And if they do, then that can end up forming tumors in other organs. And that's the problem that's associated with cancer and metastasis. So that spread. Last one. So the last one, mutations can cause change in proteins, and that can lead to a disease. So that's what this list is. All of these are genetic mutations that have led to a disease. Some of them you maybe have seen before. One of them we actually mentioned before. Remember sickle cell? Sickle cell anemia is when those cells, because the hemoglobin gene has a mutation, so the hemoglobin doesn't have a normal shape. Those cells get more like banana shaped. They don't carry oxygen very well. They burst easily. The person ends up being anemic. You can end up with crises where you end up with clots forming in deep organs. That can reduce their lifespan because it can cause liver failure, kidney failure, along with all of the painful part. But that's because of a single mutation. A single mutation that happened in a germ cell. So it happened in an egg or a sperm that then led to an offspring that had this. And that offspring passed it on to their children and their children and their children. Same thing, hemophilia. So hemo, always think blood. So a hemophiliac is somebody that doesn't make blood clotting proteins. So in your blood, you have proteins that circulate that help to clot the blood if there's damage to a blood vessel. So hemophiliacs, they're missing one or more of those proteins, all because of a mutation. So if they cut themselves, what happens? They keep bleeding. They don't clot. So they're the ones, like if they whack their arm and they get a bruise, it doesn't just stay a little bruise. They end up with this big, huge black and blue, like hematoma, like this big blood pocket that forms because they don't have clotting factors. So again, passed on from parents. So it's passed on. All of these are ones that have been passed on from parents to their offspring. You can see that some of them are really pretty severe. Huntington's disease, that one ends up leading to like complete paralysis. Terrible. Down syndrome. Down syndrome is when the chromosomes don't separate correctly during development. Causes lots of like physical impairments, heart, eye defects, sometimes cognitive, sometimes not, sometimes physical issues. Cystic fibrosis is a mutation so that you don't get good salt transport. So like your mucus is super thick. And so they're the ones that always have respiratory issues. So when they cough, it sounds like it's like mud in their lungs because they don't make enough watery mucus. Their mucus is super thick.
Does diabetic normally have something to do with the hemo? Is that why they say you have to be careful about cutting certain stuff? Like,
Oh, so like in hemophilia, you mean? Well, so it doesn't really have so much to do with diabetes. With hemophilia, if you get a cut, you just don't clot. So you just keep on bleeding, okay? Now with a diabetic, their issue is their fingers and toes are numb. So if they get a cut, they don't feel it. It's not that they don't stop bleeding. They will stop bleeding, but they don't even know that they've cut themselves. So if they don't know they've cut themselves, then they don't go and put a Band-Aid and keep antibiotic ointment on it and stuff. And so they increase risk of what? Infection. So that's really diabetes is you have poor healing, but it's not because of no clotting. It's because you have poor circulation and you don't feel it. And so that increases that risk. Okay, so I'll quit there. Just sort of an interesting, I think mutations are such an interesting connection to genes, DNA, nucleic acids. So I really wanted to try to sort of more focus on these compared to doing a whole lot about protein synthesis. So just like go over the basics of protein synthesis, right? Just sort of when I say like the basics, like go over sort of the steps of what happens, just the very basic steps, almost more like what happens here, okay? DNA is converted to RNA, which is converted to proteins, okay? But that DNA sequence, that gene sequence is really what determines what that protein is going to be. And if you have a mutation in a gene, it'll end up leading to issues from there. All right, so things that I have the take-home exam because, and you really want to make sure that you keep this